Welcome everyone to today's webinar, Illumina DNA PCR-Free Prep, Tagementation, and Introduction. Today we will be covering the following topics: advantages of Illumina DNA PCR-Free Prep, consumables and equipment, input and workflows, library prep protocol, library evaluation and quality control, sequencing, analysis, and we'll end with some resources. So first, let's discuss the advantages of the Illumina DNA PCR-Free Prep. The Illumina DNA PCR-Free Library Prep is based on Nectera BLT, or bead-linked transposome technology. In this workflow, double-stranded DNA is added to the BLTPS, bead-linked transposome PCR-free, which simultaneously fragments and adds partial adapter sequences to the DNA. After this, indexes are ligated onto the fragments, and the final library is eluted from the beads, ready for sequencing without any additional PCR amplification. There are several key features and advantages to the Illumina DNA PCR-Free Library Prep. First, it's a significantly faster workflow with 55 minutes of hands-on time and 90 minutes of total turnaround time. It supports many DNA input types, from genomic DNA to whole blood, dried blood spots, and saliva. If we use 300 or more nanograms, or if we use the integrated protocols for blood, dried blood spots, and saliva, we'll also expect normalized output. We have flexible protocol options. We have a thermocycler protocol for standard input and low input, and we also have a HIVEX protocol for standard input. And lastly, Illumina DNA PCR-Free is part of a comprehensive solution, which includes the use of unique dual indexes, a whole workflow from sample to analysis, results interpretation with TrueSight software suite, and is compatible with automation methods. Overall, Illumina DNA PCR-Free is part of a comprehensive workflow from library preparation to sequencing on most Illumina platforms, with several analysis options such as the TrueSight software suite and base app. Illumina DNA PCR-Free library prep reagents are automation-friendly with scripts developed from these vendors: Eppendorf, Tecan, Hamilton, Perkin-Elmer, and Beckman Coulter. Now we'll review the required consumables and equipment for this workflow. There are several components that need to be ordered before beginning your library prep. First, we'll need the library prep reagent kit. We have a 24 sample kit version and a 96 sample kit version. We'll also need at least one index adapter set. These are the IDT for Illumina DNA/RNA UD, or unique dual indexes, Set A or Set D. Sets C and D are coming soon. For most instruments, we'll also need to order a custom sequencing primer. We have two kits. One is the Read1 VP10 Read1 primer, and the other is the Read1 and Index 2, the VP10 and VP14 custom primer kit box. We'll discuss custom primer requirements in greater detail later in today's webinar. And then if you're also going to be working with blood and saliva input, you'll need the Illumina Lysis reagent kit. There are also several user-supplied equipment and consumables that should be acquired in advance. If you're going to be following the thermocycler workflow, you'll want to have a thermocycler. If you're following the HIVEX workflow, you'll need the HIVEX system and a plate shaker. All workflows require a magnetic stand, vortex mixer, centrifuge, and water bath. And you'll also need kappa qPCR or single-stranded qubit reagents for the final library quantification. Please consult the consumable and equipment section of the reference guide for a full list of required equipment for your selected workflow and input type. Lastly, I would like to point out a few reagents that are unique to this prep kit. First is those single-stranded qubit reagents. Because the final libraries are single-stranded and cannot be quantified by double-stranded qubit reagents, we'll need these single-stranded reagents. So qPCR quantification is an alternative to this as well. There are also the Illumina purification beads, the IPB. These are beads that are used for DNA purification and size selection of the fragments, which is based on a ratio of the beads to the sample. These beads are stored at room temperature, and freezing will damage their function. We also have the bead-linked transposomes PCR-Free, the BLT-PF, which are a unique BLT for this workflow. This is, so far, our only bead that is stored in the freezer at -20 Celsius, and this has been validated up to 6 freeze-thaw cycles. Now we will review the input and workflow options for this library prep kit. Illumina DNA PCR-Free supports 4 types of input: genomic DNA, whole blood, dried blood spots, and saliva. For genomic DNA input, we will need high-quality genomic DNA. For 25 to 299 nanograms of input, we do need to quantify that input, and final libraries will not be normalized because we'll be below the saturation point of the BLT-PF. If we use 300 to 2,000 nanograms of input, we don't need to quantify the input because we'll be saturating the beads. This will result in normalized final libraries. Some resources for the genomic DNA workflow include the protocol, which is found in the Illumina DNA PCR-Free reference guide. We also have a support page with several FAQ, and a web-based training that reviews the product overview and summarizes the protocol. For whole blood input, we will need 20 microliters of fresh whole blood in EDTA collection tubes. These will be stored at 4 degrees Celsius and must be processed within 3 days. Processing of whole blood will also require the Illumina reagent kit. And with whole blood, we expect to extract enough DNA to saturate the BLT-PF, so high yield is expected. This means input quantification is not required and final library normalization is expected. Some resources for the blood lysis workflow include the protocol, which is found in Appendix A of the reference guide. We also have the support page, FAQ. And the web-based training includes best practices on blood collection and storage, as well as blood extraction. For dried blood spots, we will need 6 3mm squared dried blood spot punches and the Whatman 903 protein saver cards. These cards will be stored at room temperature. To process these spots, we will need the Illumina lysis reagent kit. And again, we expect to extract enough DNA to saturate the BLT-PF, so high yield is expected. This means input quantification is not required and final library normalization is expected. Some resources for the dried blood spot workflow include the protocol, which again is in Appendix A of the reference guide. We have a support page with FAQ about the dried blood spots. And the web-based training includes best practices on blood collection and storage, as well as blood extraction. For the saliva workflow, we will need 50 microliters of saliva from Origene collection tubes. These will be heat stabilized for 1 hour at 50 degrees Celsius prior to room temperature storage. We will also need the Illumina lysis reagent kit, specifically the extra Illumina purification beads that are in this kit. We expect to extract enough DNA to saturate the beads, so high yield is expected. This means input quantification is not required and final library normalization is expected. Some resources for the saliva lysis workflow include the protocol, Appendix A of the reference guide, our support page, which has FAQ about the saliva workflow, and the web-based training, which includes saliva lysis best practices. So depending on our input amount, we will be following different workflows, and we may or may not expect normalization of the final library. For instance, if our input is 25 to 99 nanograms of DNA, this is considered low input. 100 to 299 nanograms of DNA is standard input, but this will be below the saturation point of the bead. 300 to 2,000 nanograms of DNA is standard input as well, but now we will be saturating those beads. And then blood and saliva are considered blood and saliva input. When we are below that saturation point of the beads, we will need to quantify that input. So pretty much anything under 300 nanograms input, we will need to quantify. In those cases, we will not have final library normalization. In contrast, when we use 300 or more nanograms or the blood and saliva workflow, we are saturating those beads, so we do get final library normalization. And in terms of the protocol, for 25 to 99 nanograms of DNA, we will follow the thermocycler low input protocol. For 100 to 299 nanograms of DNA, we will follow the thermocycler standard input protocol. For 300 to 2,000 nanograms of DNA, we will be using the thermocycler standard input protocol, or we can use the Heibeck's protocol. And then for blood and saliva, we will be using the thermocycler standard input protocol once we've done that extraction. For all input, we will need to quantify that input with a fluorescence-based quantification method, such as the qubit double-stranded DNA high-sensitivity kit. We have separate chapters in the reference guides for the various workflows that can be employed. The choice in the workflow will depend on our input amount, input type, and equipment usage. For example, the thermocycler standard input workflow will be used if our input is between 100 to 2,000 nanograms. Again, if we're below 300 nanograms, we're not saturating, so we need to quantify, and if we're above 300, we will saturate. We don't need to quantify, and final libraries are normalized. Whole blood, dry blood spots, and saliva will also follow the standard protocol after we extract the DNA following the guidance in Appendix A. And then the thermocycler standard input protocol is found in Chapter 2 of the reference guide. For thermocycler low input, so this is 25 to 99 nanograms of DNA input, this is going to be following Chapter 3 of the reference guide. And then the final workflow, the Heibeck's workflow, which again supports greater than 300 nanograms of DNA. This will be found in Chapter 4 of the reference guide. To ensure success during library preparation, our input must be clean and pure. Purity can be assessed with a UV absorbance method like the nanodrop. We'll want the 260/280 ratios to be between 1.8 and 2. And the 260/230 ratios to be between 2 and 2.2. Samples should also be free of contaminants such as EDTA, phenol, ethanol, and other organic contaminants, which are known to inhibit the Tagementation reaction and can result in either low or no final library yield. Now let's review the library prep protocol in more detail. So in general, for libraries to sequence on Illumina instruments, the DNA insert needs to be linked with sequencing adapters. Each adapter has 3 parts. For clustering, libraries need to have a P5 and P7 flow cell binding site on the far ends of the library. For sequencing, we need to have read 1 and read 2 sequencing primer binding sites that flank the DNA insert that we're sequencing. To be able to multiplex multiple samples in a run, we'll need to have a unique index or barcode sequence for our samples. For this prep kit, we have dual indexes, as indicated here. Please note that the Illumina DNA PCR-Free Prep kit generates single-stranded final libraries. Before beginning your Illumina DNA PCR-Free library prep, please be sure to prepare your consumables in advance. Prepare diluted HP3, which is sodium hydroxide, for each reaction. You'll want to also prepare 80% ethanol, fresh, for each reaction. If you're following the HIVEX protocol, you'll want to confirm that your HIVEX with its MIDI insert is preheated to 45 degrees Celsius. And if you're following the thermocycler protocol, you'll want to make sure your TAG and ELM programs are pre-programmed on that thermocycler. So here are the steps involved in the Illumina DNA PCR-Free library prep protocol. First, we're going to tagment the genomic DNA. We'll do a post-tagmentation cleanup, followed by ligating the indexes. We'll then clean up the final libraries, at which point our prep is complete. And when we're ready to sequence, we can quantify and pool the libraries, dilute to the starting concentration for our instrument, and then set up the sequencing run. No matter the source of input DNA, all input types will go through the same workflow as shown on the right. When using one of the workflows for blood or saliva, please note that extracting the DNA via the protocol found in Appendix A, the isolated DNA will be fed into Chapter 2 for preparation with the thermocycler standard input workflow. The first step of the Illumina DNA PCR-Free protocol is to fragment and tag the DNA with partial adapters, a process called Tagementation. We'll begin by preparing the Tagementation reagent, the BLT-TF, the bead-link transposon, PCR-free, and Tagementation buffer 1, TB1. We'll then incubate our 50 microliters of DNA and Tagementation reaction reagent, and then we'll run the TAG program on the thermocycler or incubate on the HIVEX. The best practices for this section include following the protocol as written. You'll want to use the preparation steps in each section to prepare all the reagents in advance and bring them to temperature. For handling the bead-link transposon PCR-free, we'll want to bring that reagent to room temperature prior to use and make sure that we fully vortex it well for complete resuspension. Make sure when it's stored to store it frozen and upright so that the beads are submerged in the buffer. And because this reagent has a high viscosity, we'll want to aspirate and dispense slowly to ensure accurate volumes. In the second step, the post-tagmentation cleanup, we will stop the tagmentation reaction and wash the DNA. We will add stop tagment buffer 2, ST2, and incubate at room temperature. We'll then wash with tagmentation wash buffer. We'll incubate, leaving that supernatant in the plate to prevent the beads from drying out, and then we'll place it on a magnet. The best practices for this section include dispensing the ST2 directly onto the beads. We will pipette Tagment wash buffer and ST2 slowly to minimize foaming since they're a bit soapy reagents, and we'll fully resuspend the beads with the Tagment wash buffer to fully wash them. In the next step, we will ligate the I7 and I5 index adapters to each sample. So first, we will add the extension ligation mix, ELM, and then we'll add our indexes for each sample. Again, these are the IDT for Illumina DNA/RNA UD indexes. We'll then run the ELM thermocycler program or incubate on the HIVEX. We'll then do a second wash with the Tagment wash buffer and elute the libraries with the diluted HP3, which again is sodium hydroxide. This causes the libraries to now be single-stranded and eluted. The best practices for this section are to make sure all of the Tagment wash buffer has been removed from the samples before adding the ELM and adapters. We'll want to aspirate and dispense the ELM slowly since it is a highly viscous solution. For the index adapter plate, each well of the plate is single use only, and we'll want to puncture the foil seal prior to use. For indexing, we'll be using the IDT for Illumina DNA/RNA UD or unique dual indexes. Each well in the plate contains a pair of indexes, an I5 and an I7. These indexes are provided in plates. Each well is single use only. And if you're going to be doing low plex pooling or only putting a few samples in a run, please consult the index adapter pooling guide to determine which index combinations are best for color balance. In short, we'll be pooling indexes by column rather than by row, but please consult that guide for further guidance. And lastly, make sure your sample sheet has 10 cycles of sequencing per index read because these are dual 10 base pair indexes. In the next step, we will size select and clean up the libraries using the Illumina purification beads. Please note that if we're doing the low input, so that's 25 to 99 nanograms, this is going to be a single-sided selection to ensure that we maximize the amount of library we get in the end. Whereas if we're using 100 or more nanograms of input, we'll be doing a double-sided size selection to have a tighter library size. We will add the Illumina purification beads to the well containing the library eluted off the BLTPF beads. We then incubate and place a plate on a magnetic stand. Next, we'll discard the supernatant and wash the beads with 2 rounds of ethanol wash. Finally, we'll elute the libraries and resuspension buffer. The best practices for this section include for the Illumina purification beads, to bring them to room temperature before use so that they fully combine the DNA. We'll want to completely resuspend those beads by vortexing frequently, especially if you're working with a lot of samples, they can settle out. We'll want to use freshly prepared 80% ethanol to get a good clean wash. And we'll want to ensure that the correct bead volume is used for each size selection step because the volume of beads will determine what size fragments are being bound, so the ratio there is very critical. At this point, our libraries are complete and ready for library evaluation and quality control. When assessing the library's quality, please note that the Illumina DNA PCR-Free libraries are single-stranded due to the elution of sodium hydroxide. Due to this, the libraries cannot be visualized on a bioanalyzer or a tape station. Since we can't see their size, we're going to be using 450 base pairs as the library size for molarity calculation purposes. We do have a tech note about adjusting that size, the tunable insert sizes with Illumina DNA PCR-Free Prep Tagementation. This tech note goes into how you can modulate that insert size between 350 and 550 base pairs. So if you are doing that modulation, you'll use that size, again, 350 or 550. But if you're following the standard workflow, you'll use 450 base pairs as your size. We have 2 methods for quantifying Illumina DNA PCR-Free libraries. When our input is greater than or equal to 300 nanograms, then we can quantify with single-stranded qubit reagents. Please note that the use of double-stranded qubit reagents will result in a value of 0 nanograms per microliter. When calculating the molarity, we'll want to use the following equation, which is based on a 450 base pair size and 660 gram per mole mass. This reduces to the equation of the molarity in nanomolars equals the yield in nanograms per microliter times 3.36. The output of this equation is the double-stranded DNA molarity equivalent. Our other method of quantification is kappa qPCR. This is required when our input is less than 300 nanograms, but it can be used for any workflow if you prefer. When calculating the molarity, please use 450 base pair size and 660 grams per mole as the DNA mass, so that this outputs the double-stranded DNA molarity equivalent. Step-by-step instructions can be found in each chapter of the reference guide. I do want to note that if you are saturating the beads, which again is 300 or more nanograms of input, we don't need to quantify the individual libraries, but since there is day-to-day variability based on your vortexing and storage of the reagents, we do want to quantify the final library pool. When you're ready to sequence, we will denature and dilute the libraries. If sequencing on the NovaSeq, guidance can be found in the reference guide. with loading concentrations provided for standard and XT workflows. Please note that we have different loading concentration values depending on if we use qPCR or single-stranded qubit to quantify our libraries. Following the low input workflow, we will only be able to sequence on the XT workflow due to the expected lower final library yield. If sequencing on the NextSeq 1000, 2000, we'll do a kappa qPCR quantification. And if we're doing onboard denaturation, the loading concentration will be 1000 picomolar. This guidance can be found in the NextSeq 1000 and 2000 sequencing system guide. If you prefer to manually denature, then we're going to use 100 picomolar final loading concentration, and that's going to be in the NextSeq 1000 and 2000 denature and dilute libraries guide. In all situations, we do want to still add sodium hydroxide per the standard protocols because this will remove any secondary structures in our libraries, even though they're already single-stranded. If we're sequencing on the iSeq 100, our onboard denaturation, where onboard denaturation occurs, we'll be loading 120 picomolar final loading concentration. This guidance is found in the iSeq 100 sequencing system guide. And then for all other supported instruments, please refer to the optimal loading concentrations for Illumina DNA PCR-Free Libraries tech note, which has those loading concentrations for other platforms. Now our libraries are ready for sequencing. Illumina DNA PCR-Free libraries are supported on all Illumina instruments except the HiSeq X. So this is the iSeq 100, the MiniSeq, the MySeq, NextSeq 500 and 550, NextSeq 1000, 2000, HiSeq 1000, 1500, 2000, 2500, and the NovaSeq 6000. Please note that most platforms and consumables will require either the Read 1 or the Read 1 and Index 2 custom sequencing primers. When preparing for sequencing, we must take these 4 factors into consideration: the sample sheet, pooling guidelines, denature and dilute, and then run setup. To create our sample sheet, please use the newest version of Illumina Experiment Manager, version 1.19, which has this prep kit already as one of the predetermined drop-down menus. For local run manager setup, we have pre-made library prep kit definition files that you can import into LRM to rapidly make sample sheets. And for NextSeq 1000, 2000 runs, base-based instrument run setup also has this library type available in the drop-down menu. When considering pooling for your run, you'll want to consider the number of samples you can do per run. To figure that out, you can consult our data sheets, application notes, tech notes, as well as the sequencing coverage calculator. And if you're doing low-plex pooling, so again, that's only a couple samples per run, we'll want to look at the index adapter pooling guide so that we could choose color-balanced indexes. When we do denature and dilute, we'll want to follow standard protocols to denature with sodium hydroxide, and then dilute to our target loading concentration, which depends on our instrument. For run setup, we'll need to physically add the custom sequencing primer or primers to the cartridge. We'll then need to instruct the instrument to use that primer, so we'll need to either check off the Read 1 or the Read 1 and Index 2 selection as is required by the instrument and reagent type. We'll also need to set up the appropriate read lengths for this library. As promised, we'll now discuss the required custom sequencing primer options in more detail. Due to how different Illumina instruments sequence Index 2, some instruments only require a custom Read 1 primer. Other instruments require both custom Read 1 and custom Index 2 primers. For this reason, we offer 2 custom primer kit configurations for purchase, which are sold separately from the library prep kit. So in this first kit in purple, this is just going to be our Read 1 primers. It's got 4 tubes of the VP10 custom Read 1 primer at 7.5 mils each. This is for use on the NovaSeq 6000 with version 1 consumables the MySeq, and HiSeq 1000, 1500, 2000, and 2500. The second kit in blue is our kit that has the Read 1 and Index 2 custom sequencing primers. So there are 2 tubes of the VP10 custom Read 1 primer at 7.5 mils each, and 2 tubes of the VP14 custom Index 2 primer at 10.5 mils each. This kit is for use on the iSeq 100, MiniSeq, NextSeq series, HiSeq 3000, and HiSeq 4000. Please note that these custom primer kits are sold separately from the library prep kit, since it does depend on what instrument you'll be sequencing on. These custom primers are cocktail, so they are compatible with Illumina DNA PCR-Free libraries, SciX, and any other Illumina library. These custom primers are provided at the final concentration and require no dilution, hence the large volumes, so you'll just put them directly into the cartridge. And lastly, please note that custom primers are not required for the NovaSeq 6000 version 1.5 sequencing reagents. These newer reagent kits already contain the primers by default in the cartridge. We've created a resource to help clarify which primer to use, how much to use, and where to load it for each instrument in our bulletin, the Custom Primer Requirements for Illumina DNA PCR-Free Prep Tagementation Kit. So this bulletin has a lot of information, but I did just want to walk you through a couple of examples to orient you. If you do have any questions after reviewing it on your own, please feel free to contact technical support and we can help you out further. So in our first example, if we're going to be sequencing on a MySeq, we'll want to order the custom primer kit that ends in 96. Here we'll take 600 microliters of the VP10 Read 1 primer and put it into well number 18. For the MySeq, we don't need an Index 2 primer. If we're sequencing on the NextSeq 500 or 550, we'll purchase the custom primer kit that ends in the numbers 97. We'll take 2 mils of the VP10 Read 1 primer and put that into position number 7. And we'll take 2 mils of the VP14 Index 2 primer and place that into position number 9. On the NovaSeq, if we have version 1 sequencing reagents, we'll purchase the custom primer kit ending in 96. For SP, S1, and S2 flow cells, we'll take 2 mils of the VP10 Read 1 primer. And for S4 flow cells, we'll take 3.5 mils of the VP10 Read 1 primer, and we'll put that into position number 5. On the NovaSeq, we will not need a custom Index 2 primer. And if we're on the NovaSeq with version 1.5 consumables, custom primers are already in the cartridge, so we don't need to add any in addition. The second table in this bulletin provides guidance on how to instruct the instrument to use that custom primer we've added, as well as the recommended read links for the sequencing kit. So again, going over a couple of examples here. If we have a MySeq on control software version 3 or higher, this means that the MySeq is using Local Run Manager for run setup. Here, we can download the library prep kit definition file and import it as a custom library that's available in the dropdown. We'll want to make sure that we manually check off that little box for the custom Read 1 primer so that it pulls the primer we added to the cartridge. The read length will be 151, 1010, 151. For a MiniSeq on Control Software version 2 or a NextSeq on Control Software version 4, both of which use Local Run Manager as well, again, we'll download that library prep kit definition file specific to those instruments and import them as custom libraries. Here, we'll have to manually select custom Read 1 and Index 2 primers, the 2 checkboxes. And due to the number of cycles in those reagent kits, we need to set the read lengths to 149, 1010, 149. On a NovaSeq with version 1 reagents, we don't need a sample sheet to actually start the run, only for demultiplexing. So to actually just start the run, all we need to do is manually check off the custom Read 1 primer in advanced settings and make sure to set the read length to 151, 1010, 151. For NovaSeq with version 1.5 consumables, again, we don't need a sample sheet for run setup. You will need one for demultiplexing, but for the run setup, you will not need to select any custom primers because, again, this newer kit of reagents has the primers built into the cartridge. The read length will be 151, 1010, 151. I do want to point out that you must ensure to both physically add the custom primer to the cartridge and make sure you instruct the instrument to use that custom primer through the chart listed here. Otherwise, we will result in unrecoverable run failure. Now that our libraries are sequenced, we're ready for analysis. If the data was generated using the NovaSeq, then we can use the TrueSight software suite for analysis and interpretation. Data from any instrument can also be analyzed in our base-based sequencing hub app. TrueSight software suite is designed for translating sequencing data into meaningful, interpretable results in rare disease cases. It can perform secondary analysis, the comprehensive rapid variant calling, interpretation and reporting. It has case management, and it's an enterprise informatics with a holistic infrastructure and software suite. For more information, please contact our software and bioinformatics team within Illumina Technical Support. Illumina DNA PCR-free data can also be analyzed with base-based apps. We have a variety of apps available, whether you aim to sequence small, large, or human genome. If you need help selecting an appropriate app, please contact our software and bioinformatics team within Illumina Technical Support. Now I'd like to provide you with some resources and links for the Illumina DNA PCR-Free Library Prep Kit. The first is the protocol, which is going to be the Illumina DNA PCR-Free Library Prep Tagementation Reference Guide. We also have a data sheet with some data about this library prep kit, as well as several technical and application notes. The optimal loading concentrations for Illumina DNA PCR-Free has the recommended loading concentrations for most non-NovaSeq instruments. The high-performance whole genome sequencing of Illumina DNA PCR-Free Prep Tagementation tech note reviews the benefits of a PCR-free library and also reviews the number of samples per flow cell for human samples on the NovaSeq. Suitable insert sizes of Illumina DNA PCR-Free Prep Tagementation reviews how to modulate that insert size between 350 and 550 base pairs if you want something different than the standard 450 base pairs. Microbial whole genome sequencing of Illumina DNA PCR-Free Prep Tagementation technote reviews sequencing microbial libraries with varying GC content and shows how we can get good coverage even when we have high GC, such as some bacterial species. Balancing sample coverage for whole genome sequencing reviews strategies to manually balance library pools for more even representation of each sample. Here's a link to our support page for this library prep kit, a link to the downloadable library prep kit definition files for importing into Local Run Manager, and a link to the online training. Here are links to the system guides for instruments that support Illumina DNA PCR-Free libraries. And here are links to the custom primer guides and the denature and dilute guides for instruments that support these libraries. For additional resources, so for indexing, we've got three main documents. The first is the Illumina Adapter Sequences document. This has all of the sequences of the indexes for any Illumina library. The Index Adapter Pooling Guide reviews which indexes to choose for low-plex pooling runs, so to ensure that your indexes have good color balance. It's also a good resource for seeing the actual plate layout of which index goes in which well. Indexed sequencing on Illumina systems reviews how sequencing is performed on our different instruments, and it goes into more detail about why some instruments need that custom Index 2 primer, whereas other instruments do not. We also have several bulletins related to this library prep kit. The main one is that custom primer requirements for the Illumina DNA PCR-Free Prep Tagementation kit that I reviewed a couple slides back about the custom primer requirements. We also have bulletins on the bead types and Illumina library preparation kits, Illumina library prep kits and associated index kits to see if your indexes are cross-compatible, the Illumina adapter portfolio, what sequences do I use for adapter trimming, and sequencing primer compatibility of Illumina libraries and recommended library types for the iSeq 100 sequencing platform, as this prep kit is newly compatible with the iSeq 100 due to its support of custom primers. Here's a link to the sequencing coverage calculator. Thank you so much for taking the time to join me today. I hope that this session has been helpful to you.